Greetings. Thank you all for joining us. My name is Matt Hubeck. I'll be the moderator for today's webinar. If during the webinar you have any viewing difficulties, please hit F5 on your keyboard to refresh the webinar window. The audio for today's webinar will be coming from your computer, so please make sure you turn your volume up. Also, at any time during the webinar, feel free to submit questions for our presenters. After the presentation is completed, we'll address your questions in a short Q&A session. With that, I'll turn things over to Tim Moeller so he can get started. Tim?
Hello, and thank you for this opportunity to speak to you. My name is Tim Moeller, and I'm from Celsus in Vitro Technologies. I will begin today's webinar by discussing in vitro tools for drug-drug interactions and toxicity studies. I will begin with a quick overview of in vitro testing in drug discovery and development stages, as well as defining two key experimental systems for such purposes. I will then provide some examples of their applications to screen for induction, inhibition, and toxicity potentials. We cannot know where we're going unless we know where we've been. This graph created by Kola and Landis highlights this fact. In 1991, the primary reason for failure of a drug was due to pharmacokinetic and bioavailability issues. By the year 2000, this was reduced to 10% as compared to the other factors. In large part, the knowledge and understanding of the mechanisms affecting PK helped develop in vitro screening regimens to better predict in vivo bioavailability. This same approach may be able to help other areas like clinical safety and toxicology. This slide is my attempt to distill a PharmD course into 2 minutes, so here we go. There are two basic groups of enzymatic activities that aid in the metabolism and clearance of both endogenous and exogenous compounds. First is phase 1, which relies on chemical changes such as oxidation and reduction to make the compound more polar through reactive site. Phase 2 encompasses the enzymes which conjugate a chemical moiety like glucuronide or sulfate to a compound or phase 1 metabolite to increase its solubility, aiding in its excretion from the body. The pie graph to the right shows the proportion of drugs on the market in 1999 that are metabolized by phase 1 and phase 2 enzymes. For phase 1, cytochrome P450 enzymes, also known as CYPs, are the most important family for the metabolism of drugs, of which CYP3A is responsible for the lion's share of the drugs on the market. For phase 2, uridine 5-diphosphoglucuronylyl transferase, or UGT to its friends, is the most important of the conjugating enzymes in humans. Note that drugs do not need to proceed through both phase 1 and phase 2. It may be metabolized by phase 1 enzymes and then excreted, or conversely, by only phase 2 enzymes and then excreted. As well, some drugs may not be metabolized at all but excreted through transporter interactions. But of course, this is for another webinar. In terms of metabolic capacity, the liver is the most important organ in the body. 70% of the blood flow comes directly to the liver from the intestines, increasing the liver's importance for metabolizing ingested food, products, and drugs. Therefore, liver metabolism is the primary focus for studying a drug's metabolic stability, toxicological properties, and drug-drug interactions through in vitro means. Though the use of subcellular fractions like microsomes and S9 have been employed for decades, the use of hepatocytes have increased because they better represent the liver physiology, which I will discuss in coming slides. I present here a simple list of various applications that employ hepatocytes for microsomes. In many cases, either systems may be utilized. Choosing one over another will depend on your specific needs. In the case of inhibition, microsomes are the gold standard due to the simplicity and direct exposure of the drug and probe substrate. However, if transporters are involved in the uptake or drug into the cell, hepatocytes may provide a better representation of drug's inhibition potential as observed in the clinic. Other applications, such as induction, can only be performed in hepatocytes and not in microsomes due to the need of transcriptional and translational machinery found in whole cells. This is here a picture of a culture. The image is taken from a 20x HMC optics. You can see distinct cuboidal cells forming a monolayer at nearly 100% confluence. The divots are the nuclei, while the shiny bubbles are fat droplets. Drug-drug interactions are a particular concern of safety of patients on multiple medications. I'm sorry that this is a repeat. But they also can be affected by food and nutraceutical products. The result can be an increase and decrease of the elimination of secondary drug, rendering it ineffective and potentially toxic. Induction is the ability of the drug to increase metabolic capacity through transcriptional and translational mechanisms. Subsequently, the increased metabolism may eliminate a secondary drug before it produces its intended therapy. For example, the bioavailability of squanavir, the retroviral medication used to treat HIV, can be reduced by 80% if the patient is co-administered rifampicin, an antibiotic and known inducer for CYP3A4. On the other hand, inhibition is a process that reduces metabolic activity which slows the clearance of secondary drug, extending its therapeutic activity and potentially causing toxicities. Fluoxetine, also known as Prozac, is an antidepressant. It is also a potent inhibitor of CYP2D6. Deciperine, too, is an antidepressant, but also works through different mechanisms than fluoxetine. If taken together, deciperine plasma concentrations can be 4 to 5 times higher than if taken alone, and can result in dangerous arrhythmia called QT prolongation, or increase the risk of suicide in some patients. These are examples of data derived from an induction study where the induced CYP activity is compared to the basal activity. In the left panel, rifampicin is a known 3A4 inducer, represented here by blue circles, and elicits a bell-like curve. The lower induction of the highest concentration can be explained by cytotoxicity, inhibition of CYP3A4, or suppression of the transcriptional and translational mechanism. In this case, the viability, shown here in aqua triangles, is unchanged and indicates no cytotoxicity. Further investigation is needed to determine if this is an inhibition or suppression event. From literature, it can be shown that the rifampicin is an inhibitor of 3A4 at high concentrations, and therefore inhibition is the probable reason for the lower induction observed at higher concentrations. In the right panel, lansoprazole was used as a positive control for CYP1A2 induction, another critical marker for drug-drug interactions, which is represented here by red squares. It too shows reduction of induction in higher concentrations, but with reduced viability as well. This may be interpreted that the lower induction at higher concentration was due to cytotoxicity and not other mechanisms. Inhibition is the reduction of enzymatic activity through reversible and irreversible interactions. I will not attempt to distill a semester of biochemistry in one slide, but I'd like to highlight the differences using microsomes and hepatocytes in determining inhibitor concentrations, known as IC50s, of two compounds. In microsomes, CYP enzymes are freely exposed to inhibitor and probe substrates, making a direct assessment of an IC50 for a test compound. Hepatocytes, however, have a cell membrane along with transporters and other intracellular compartments that may affect the intracellular concentrations at the enzyme site. Though confounding the direct inhibitor-enzyme interaction, the use of hepatocytes may be more predictive to the drug's clinical inhibition potential than microsomes. In the case of propranolol, inhibition of CYP1A2 was more potent in microsomes than observed in hepatocytes. This may be due to lysosomal trapping, which reduces the intracellular concentration of inhibitor, causing an apparent increase in the IC50. Conversely, verapamil's IC50 for CYP3A4 is lower in hepatocytes than observed in microsomes due to uptake and an increase of intracellular concentrations of verapamil. As observed in recent publications, there is a growing focus on vetting the use of hepatocytes to predict inhibition and to explain the mechanisms involved. The liver's role in metabolism and clearance, endogenous and exogenous compounds, increases its susceptibility to toxicity from the parent compound and its metabolites. Drug-induced liver injury is such a concern that the FDA has published a guidance for industry outlining the monitoring and the liver injury during the drug development and after market release. The consequences can be dire, as in the cases of triglyceridone and bromelain, which cause liver damage, liver failure, and a number of deaths, resulting in their removal from the market. In other cases, the liver toxicity may only warrant label changes to warn of potential liver dysfunction, as in the case of Augmentin. Hepatocytes in culture can be used to monitor cytotoxicity, which may indicate the need for further investigation in clinical studies. Shown here on the left is the concentration response curve for phenylmercuric acetate, which was derived from a 40-hour exposure to hepatocytes in a 1536 well format. The plot on the right shows the consistency and reliability of such a format for measuring hepatotoxicity. From this system, high-throughput screening may be used to rank order hepatotoxicity potential and may be a warning for future in vivo outcomes. In summary, I have presented several in vitro systems that may be employed during drug discovery and development in order to assess the clinical risk of bioavailability and drug safety of potential drugs. The use of human-derived subcellular fractions and primary hepatocytes provide a safe and easy method to obtain human-specific data prior to clinical trials, which can reduce clinical failures and patient injuries. These platforms are amenable to high-throughput screening, which fits into the current strategies for pharmaceutical discovery and development. Next, Tracy Warzala will present the use of luminescent substrates for monitoring CYP activities.
Hello, everybody, and thank you for joining us today. Before I jump into my presentation, I wanted to first mention a few things about Promega. We're located in Madison, Wisconsin. We were founded in 1978 by our CEO, Bill Linton. We currently have over 2,000 products in our portfolio, which consist of reagents and instrumentation for research in genomics, proteomics, and cell analysis, as well as molecular diagnostics and human identity. We are widely known for our bioluminescent glow assay platform for cell analysis research, and today I'll be talking about one of those assays that comprises that platform, our Promega P450 glow assay, and how we can use that assay for cell-based and biochemical P450 profiling. Before I talk about the assay, I wanted to first talk a bit about bioluminescence and what it is. Bioluminescence is a form of chemiluminescence where the light-yielding reaction is derived from a naturally occurring process, such as an enzymatic reaction found in luminous fireflies, jellyfish, bacteria, and other organisms. Our chemistries primarily use firefly luciferase, which is derived from the Photinus piralis beetle shown here on the slide. Light is generated by the conversion of luciferin, which is the firefly substrate, to oxyluciferin in the presence of ATP, oxygen, and magnesium, as shown on the reaction schematic. And we take advantage of this, the fact that the components of this reaction can be measured individually, and we use this fact to develop different platform technologies for our bioluminescence assays. So there are some differences between bioluminescence and fluorescence, which is also another technology that's commonly used. Main distinction is the means by which the excited states for photon emission are created. As you can see on the left-hand side of the screen, fluorescence assays require a large influx of photons in order to create the excited state. Filters are typically required to differentiate the light that's coming into the system from light going out. This high influx of photons can often lead to higher backgrounds, largely due to a few factors. One, the need for a detector to distinguish between the excitation and the emission, and also interference from weak fluorophores that are typically present in biological samples. Sensitivity sometimes can be decreased because there is most often an overlap in the excitation and the emission spectra, as shown on the bottom. Luminescence, on the other hand, shown on the right-hand graph, uses chemical energy to absorb by the luciferin molecule to create this excited state intermediate. Therefore, there's minimum background because there are no photons that are required to be input into the system to create this excited state, resulting in a more sensitive assay. So now I'll speak a little bit about our assays. As mentioned previously, the light from our reaction is proportional to the concentration of any of the components. that are in that reaction. And we take advantage of this by developing different assays that can measure each one of these components individually. Shown here on the screen, you'll see 3 examples of ways that we use this luminescent reaction. The first reaction, shown on the top, is an example of measuring luciferase. This is our common reporter technology, where we hold the ATP and the luciferin constant. Bright glow is an example of one of our reporter technologies. The second reaction, which I'm going to be talking a lot more about today, is an example where we can measure luciferin accumulation in an assay. And we do this by holding the ATP and the luciferase constant. P450 glow is an example of this way of measuring a reaction, as well as cast-based glow, which measures apoptosis. And lastly, measuring ATP is a possibility by holding luciferin and luciferase constant. And cell titer glow is our gold standard assay for measuring cell viability. You'll notice I make reference to ultraglow luciferase in the second and third reactions here. Ultraglow is an engineered recombinant firefly luciferase that enables glow luminescence, which is typically gives a half-life on the order of 2+ hours. Some advantages of ultraglow luciferase: it has increased thermal stability, pH tolerance, as well as detergent tolerance. And all of these combined create for a reagent that is highly reduces the incidence of false positives due to luciferase inhibition. So now on to our assay. The P450 glow assay is a luminescent method to measure cytochrome P450 activity. We utilize proluciferin probe substrates to detect P450 activity. A probe substrate is commonly used to detect enzyme activity by measuring the product of an enzyme reaction on that substrate. All of our assays can be used with cell-free CYP inhibition study experiments. Many of the assays can be used for cell-based CYP inhibition studies and induction studies, which I'll be talking about later in the presentation. As you can see on the screen, the assay is a 2-step assay. The P450 reaction, which is the first step of the assay, utilizes a substrate that is a derivative of luciferin. The derivatives contain blocking groups that are recognized by the various P450 enzymes. In the first step of the assay, the blocked proluciferin substrate, which itself is inactive, is converted by the cytochrome P450 enzyme to a D-luciferin product. The D-luciferin, which is the active form of luciferin, is then detected in the second step of the assay, and this is the luciferase reaction. This part of the assay is accomplished with the addition of what we call the luciferin detection reagent. So with this assay, the amount of P450 activity that you have in your reaction is directly proportional to the amount of light that you're seeing from your reaction. And that's shown on the bottom left-hand curve. This is a D-luciferin titration curve, so the more luciferin that you have in your reaction, the more light output. Conversely, on the right-hand side, this is an example of what an inhibited enzyme reaction would look like. This is a sulfophenazole titration. Sulfophenazole is a known 2C9 inhibitor. So at higher concentrations of sulfophenazole, you'll see a decreased light output. Low concentrations to no concentration of sulfophenazole, you'll see a high output of light. We have a number of proluciferin substrates that are available for P450 studies, but we also have other enzymes substrates that are for enzymes involved in drug metabolism and modifications such as monoamine oxidase and UGT GST. For some of the P450 enzymes, including 1A2, 2C9, 2C19, 2D6, and 3A4, we sell complete screening systems, which include the human recombinant enzyme, the substrate, an NADPH regenerating system, appropriate buffers, and the detection reagents. And then for our other assays, we provide the detection reagents and the specific substrates for the enzymes. Some of the substrates are more selective than others, and I'm going to go into that in a little bit more detail in the next slide. So on the left-hand side of the screen are some representative bar graphs showing substrate selectivity for the various proluciferins available. These data were generated with recombinant enzymes. The more bars that you see on the graph, the more enzymes react with that particular substrate. For example, the luciferin H substrate that you see in the middle there is currently our most selective substrate. The chart on the right shows the substrate structures. The arrow on each substrate structure indicates a site of modification by each CYP where the blocking group is located. The indicated substrate is provided with the kits for the CYP shown in bold, but you can also use these kits for the other assays that are shown here. Our substrates for cytochrome P450 3A4, or the 3A family in general, have evolved over time. We now have 4 distinct substrates available for the 3A enzymes. Highlighted here are 2 of our most recent substrates for 3A. These are our most selective and are least affected by DMSO. The substrate on the top, our luciferin IPA, is our newest substrate and is currently the most sensitive and selective for 3A4 applications in particular, including cell-free and cell-based assays. And this is a substrate that we're going to be highlighting later in the presentation when Brad Larson from BioTek is speaking. The bottom graph shows a second substrate we have available, luciferin PPXE, which also works very well for 3A4 in cell-free systems. And it's also very DMSO tolerant. But it also reacts somewhat with the 3A5 and the 3A7 enzymes. So this substrate in particular we would recommend for anybody wanting to look at these other enzymes in addition to 3A4. The P450 glow assays compare very favorably to traditional chromatography-based assays. Shown here is a comparison showing traditional HPLC data looking at testosterone conversion by 3A4 to 6-beta hydroxylase compared to our luminescent well-based assay on the x-axis. This is our luciferin IPA 3A4 assay. You'll notice that the results show excellent correlation. These are IC50s reported in micromolar between our assay and the selective HPLC assay. Also worth noting is that the results from the recombinant system on the left match very well with the microsome-based system on the right. This highlights the selectivity of our luciferin IPA substrate. Cell-based assays have become an invaluable resource in research by allowing for data generation in the context of a biological system. The P450 glow assays can be used in a cell-based format for induction studies, which is their primary application, but they can also be used for inhibition studies. They can be used with common cultured cells as well as fresh and cryopreserved hepatocytes. What you see on the screen is a cartoon schematic showing the use of our 3A4 selective luciferin IPA substrate that I spoke about just now. When added to the cell culture, the substrate is able to cross the cell membrane, so it is cell permeable. If present and active, the 3A4 enzyme converts this proluciferin substrate to the active form of luciferin, and that active form of luciferin is able to cross the membrane into the surrounding medium. The addition of the luciferin detection reagent, which contains luciferase and everything else needed to generate light, is then added to the well. Light generated is proportional to P450 activity. And being a cell-based system, not a recombinant one, there are many CYP enzymes that can be present in the cell-based system that might interact with the various substrates. And for this reason, we have a select number of the P450 glow substrates that we recommend for use in the cell-based system, and those are shown on the screen here. So our most selective substrates for cell-based induction applications in particular are those for 3A, which is seen with the luciferin IPA on the top, 2C9, which is luciferin H, 1A2, which is the luciferin 1A2 substrate, and as well as the 1A family, which is represented by luciferin CEE. The cell-based P450 assays can be run in 2 modes, lytic and non-lytic. The general workflow for each assay method is depicted here. Following a treatment period, P450 glow substrates are added to cells in culture and incubated. The luciferin detection reagent itself lyses cells when added directly to the culture. So when running the assay in a lytic mode, as shown on the left, light is generated from the P450 reaction after a 20-minute incubation. The P450 glow assay can also be performed in non-lytic mode and multiplexed with a cell viability assay like cell target glow. The advantage of multiplexing with the viability assay is that the cell health can be monitored as an effective drug treatment. Following substrate addition for the non-lytic method, a portion of the media is removed from the test well to a second assay plate. As you recall from the previous slide, the luciferin that's generated within the cell is cell permeable and can be released into the medium. So the P450 glow assay is performed in a secondary plate with a portion of the assay medium that you remove from your cell-based plate. You can also assess cell viability then by adding cell titer glow reagent, which is a reagent that lyses cells and measures ATP directly to the cell culture plate. So now you can normalize your P450 activity to the viable cell number from cell titer glow. Shown on the screen is some representative data showing the use of our luciferin 1A2 substrate for 1A2 induction studies. This was performed in a cell-based system with cryopreserved hepatocytes. You'll notice that over the course of time, you see an increase in light due to an induction of P450 activity due to the treatment of omeprazole, which is a known 1A2 inducer. It's also possible to multiplex the P450 glow cell titer glow combination previously described with a third fluorescent readout to form a triplex assay that contains both luminescent and fluorescent probe substrates. Multiplexing several different P450 assays together is a very valuable tool for evaluating potential drug-drug interactions. After treatment with a test compound, as shown here, the luciferin IPA substrate for 3A4 is added to culture, and a portion of the culture media is removed to a second plate where the P450 glow 3A4 assay is performed. Next, a fluorescent CYP1A substrate, in this case ethoxyresorufin, is added to the remaining cells in culture, followed by incubation and treatment and measurement of fluorescence. Fluorescence is directly proportional here to the amount of CYP1A activity present in the well. And then lastly, as described previously, you can quantify the number of viable cells in the well by adding cell titer glow directly to the cells left in culture. Shown on the screen is some representative data showing the triplex assay in action. On the left-hand side of the screen is a rifampicin titration. As Tim mentioned, rifampicin is the known 3A4 inducer. And here you see a dose-dependent induction of 3A4 as measured with the P450 glow assay. No effects of CYP1A as measured with the fluorescent assay or viability measured with cell titer glow were noted here. On the right-hand side of the screen, you'll see an omeprazole titration, which shows a different profile but an expected profile. Biphasic responses are common when studying CYP induction. You see CYP induction at lower concentrations but inhibition at higher concentrations. And as previously shown, omeprazole is a known 1A2 inducer, and you can see that here with the fluorescent CYP1A assay. You'll see induction up to approximately 100 micromolar. Past that, the activity starts to drop and you begin to see inhibition. Looking at the cell viability result with cell titer glow, you see that cells show a decreased viability over the course of treatment with higher doses, and that decrease coincides with the reduced expression of CYP1A, cluing in that cytotoxicity is playing a role here, but enzyme inhibition is the main culprit. When you look at all the assays together on this one graph, it's hard to see what's going on with 3A4, but when you zoom in on the 3A4 result, as shown on the bottom graph, you see that over the course of treatment with omeprazole that the 3A4 activity also appears to decrease. And this makes sense because omeprazole is a 3A4 substrate. The more omeprazole present in the system, the more it competes with the 3A4 IPA reaction. So you see a decreased activity due to competition and inhibition. So in summary, luminescence provides the following benefits: low background, which enables greater sensitivity and dynamic range. We don't have any fluorescence interference with our system. P450 glow can be performed in both the biochemical and a cell-based format. And if you're interested in multiplexing the cell-based with cell titer glow, you can certainly do that if you want to evaluate your cell health effects due to treatment. And hopefully you've realized that the assay is very easy to perform. The ADME-Tox assay procedure is easily adaptable to both manual and automated processing. With that, I'm going to hand this over to Brad Larson, who's a principal scientist at BioTek, who will describe their efforts to combine the cell systems and assays described here today for automated P450 profiling applications.
Thanks, Tracy. So again, before I begin, I'd also like to thank everybody for joining us with this webinar here today. So what I'd like to do, as Tracy just mentioned, is to continue to build on the story that you've been hearing from Tim and Tracy. So just to begin, so as most, if not all of you know, ADME-Tox testing continues to move further upstream in the drug discovery process. Therefore, more and more compounds need to be tested to generate a safety profile for the compounds. This has created the need for higher throughput in vitro assay chemistries that can be automated using appropriate instrumentation and use appropriate models to generate the most in vivo-like data. Here I'll be showing you 3 case studies where we combine these 3 to create simple, efficient processes that are robust and deliver appropriate pharmacology. So to begin with, I'll start by discussing how the instrumentation that was incorporated into these projects can be used to dispense the cells, reagents, and other assay components. Then I'll explain in greater detail the 3 case studies which I mentioned earlier, which include biochemical as well as cell-based CYP inhibition and induction profiling and multiplexing applications using recombinant CYP enzymes, liver microsomes, or hepatocytes. So the first instrument that was used is our precision microplate pipetting system. It combines an 8-channel pipetting head and as well as a 8-channel bulk reagent dispenser. And for these applications, it was used to titrate compounds in 96-well format and then transfer those compounds from the cell plate to either 96 or 384-well assay plates. And then also for low to medium throughput applications, the instrument can also be used to dispense cells, media, and other assay reagents. The reason why it can do this is because the instrument has a small footprint, which allows it to be put into existing laminar flow hoods, which allows for easy sterile manipulations with the use of sterile disposable tips. The next instrument is our MultiFlo microplate dispenser. It combines up to 2 8-tip non-contact peristaltic pump dispensers and 2 variable tube syringe pump dispense manifolds. And it was for 2 of the projects. It was also used to dispense cells and media as well as reagents. It does this through non-contact dispensing, and it's capable of dispensing into 96, 384, or 1536-well plate formats. And through chemical or autoclave sterilization, you can ensure sterile manipulations of your cells and media. And it also has a small footprint, so if you are using it offline, it can be easily, again, put into existing laminar flow hoods to ensure running assays in a sterile format. And then finally, our EL406, which is a combination washer dispenser. This combines the wash manifold with the 8-tip non-contact peristaltic pump and syringe pump manifolds, which I just previously described. So again, you can accomplish non-contact dispensing of cells and media in a sterile format. Plus, with the addition of the wash manifold, it's capable of removing media from the wells during media exchanges due to the fine control of the Z-axis that we have for the manifold, which allows you to easily remove a majority of the media from the wells without disturbing the cell layer at the bottom of the well. And in this way, you can accomplish full plate media exchanges in less than a minute. The manifold can also be chemically sterilized to ensure that this is run in a sterile format. Again, the instrument has a very small footprint, so if it's not being used in an online setting and you don't need to run many plates for your experiment, it can easily be put into a laminar flow hood again to ensure sterile manipulations. And then finally, at the other end of the assay process, we used a number of our Synergy microplate readers to detect either the luminescent or fluorescent signals that are used in the assay chemistries that were just described by Tracy. Each are capable of fluorescent, luminescent, or absorbent detection without having to change out the inner workings of the reader. The Synergy MX on the left is a monochromator-based instrument which captures fluorescent signals using variable band passes as well as having a separate luminescent detection. And then the Synergy H1 and H4 are hybrid readers which incorporate not only the monochromators, which I just spoke to you about, but also filter-based detection in the same unit, which gives you the flexibility to quantify signal using either detection mode. So I'm going to switch gears now, and I'll start talking about how we have combined the various cell and enzyme models, assay chemistries, and instrumentation. The first case study looks at the ability to profile various compounds against multiple recombinant CYP enzymes in a biochemical format. The automated assay, which incorporates the P450 glow assay systems that you just heard about, is performed in the following way. This was done, as you can see, using our precision dispenser. The compounds are titrated and then added to the wells of the assay plate, followed by addition of an P450 enzyme, substrate, and potassium phosphate solution. Then followed by NADPH regeneration system addition, which initiates the P450 reaction. The wells of the plate are incubated at room temperature for an appropriate time depending on the substrate that you're using. The luciferin detection reagent is then added. The plate's mixed and incubated for 20 minutes to allow the luminescent signal to stabilize, and then the luminescent signal from each well is detected using the Synergy H4. Z-factor, which is a measure of assay robustness first described by Zhang in 1999, takes into account the difference between the averages of a positive and negative control as well as the variation around each mean. An ideal assay would have a z-prime value of 1.0, and an excellent assay is described as having a z-prime value of greater than or equal to 0.5. Because of that, here we used the z-prime experiments to validate the robustness of the automated assay procedure that was just described. For this case study, we examined 3 CYP isoforms, CYP2C9, 2D6, and 3A4. And as you can see, the z-prime values for each automated assay are above 0.8, which is indicative of a highly robust assay procedure. After a successful validation test, we then went on to perform the compound profiling. 9 compounds in total made up the profiling panel and were run with each of the 3 assays. The graphs above show the inhibition curves that were generated for all the compounds tested with each assay. And then below on the left, IC50 values that were determined from the curves for the known inhibitors of each of the 3 isoforms are listed. And on the right, previously generated literature IC50 values are also included. By comparing the values generated with the automated assay to those previously generated, you can see that there's close agreement between the 2 values, which further validates the automated assay procedure and its ability to provide appropriate inhibitory data for lead compounds. The second case study once again looks at CYP inhibition profiling. However, the goal here was to determine whether the differences would be seen in compound IC50 values generated using human liver microsomes in a biochemical format compared to those generated when using human hepatocytes in a cell-based format. The reason, again, as previously explained by Tim, is that there has been a recent push by ADME-Tox researchers to generate CYP inhibition data using cells instead of a more purified enzyme format because, again, as was previously mentioned, the cells contain the full complement of enzymes which can affect the availability of the compound to the cytochrome P450 enzymes. So our goal here was to validate whether or not this is truly a valid hypothesis. As you can see, the two assay formats have a very similar procedure to what I previously described. Each begins by, again, titrating the compounds and adding them to the wells. This assay was performed at 37 degrees instead of room temperature. So before adding anything else to the wells, we incubated the plates for about 20 minutes to allow the compounds to warm to 37 degrees. We then added either 5 microliters of microsomes and NADPH or 10 microliters of suspension hepatocytes to the wells, and followed by a pre-incubation period that you see. The CYP reaction was then initiated through a 5 microliter addition of various substrates, and then followed by incubation using an appropriate time, again, depending on the substrate being used. Detection reagent was then added, followed by the 20-minute incubation time that Tracy described previously, and then the luminescent signal was again detected using the Synergy H4. Once again, Z' experiments were performed to validate the automated microsome and hepatocyte-based assays for each of the 3 CYP enzymes included in the study, which here was CYP2C9, 3A4, and 1A2. And again, as you can see, the Z' values, which range from 0.7 to 0.8, confirm that the assay, enzyme, or cell model and instrumentation combine together in a robust fashion that delivers consistently repeatable data. Following the Z' validation, inhibitor profiling was again performed. This was done to not only further validate the assay, but as mentioned previously, to examine whether differences would be seen when the compounds were run with the 2 assay formats. 12 compounds were tested in dose-response format, and the process was repeated 3 times with each assay to ensure repeatability of the data. Inhibition curves and IC50 values for a single known inhibitor tested with each assay are shown here. Similarities of the curves, as well as the low variation within the average IC50 values, show the repeatability of the process using automation. And then here we see data from a single run that was generated using the automated process for the biochemical microsome-based assay as well as the cellular hepatocyte-based assay. This helps to further illustrate how multiple compounds can be easily and efficiently profiled using this procedure. Then here we see the average IC50 values that were generated from the 3 runs performed for the known inhibitors of each enzyme isoform. Values for the microsome-based assay are shown on the left, and values for the hepatocyte assay are on the right. Literature IC50 or KM values are also shown. By comparing the results generated by the automated assay to those previously generated, you can see that for the majority of the compounds, there's close agreements between the values. However, there are differences that are seen between IC50 values generated with the 2 assay models. The yellow highlighted cells shown here in the 2 tables show IC50 values that differ by roughly a factor of 10 between the 2 formats. And then the graph that's shown here further illustrates this point. You can clearly see how the inhibition curves differ between the 2 formats for the compounds included, which lead to the differing IC50 values. One reason, again, as Tim alluded to earlier, is explained is that hepatocytes, again, contain the full complement of enzymes, such as transporter enzymes, which can potentially change the concentration of a compound available to act upon the P450 enzyme. This helps to prove, again, why CYP inhibition assays need to be run in the most in vivo-like format. The final case study looks at the ability to detect CYP enzyme induction by lead compounds. The assay procedure specifically examines the activity of two important CYP enzymes, CYP1A and 3A4, that have been shown to be inducible by compounds in a cell-based format using cryoplateable hepatocytes is what we used here. What's unique about this procedure, again, this is going to kind of continue to build on what Tracy told you earlier, is that in this procedure, the activity of both enzymes are measured in the same assay well and also includes a cell viability measurement. So therefore, by measuring these three components in the same well, the user can reduce assay variability and reagent costs and generate a more in vivo-like data. The 96 and 34 well triplexed assays, which are what we tested here, are performed in the following manner. You add the cryopreserved cells to the wells of the assay plate and incubate overnight at 37 degrees. On day 2, spent media is removed. This is all performed by the EL406, as you can see, and fresh media is replaced onto the cells. On day 3, spent media is again removed, and the previously titrated compounds are overlaid onto the cell layer. This creates a 48-hour dosing period. And then on day 5, the media again is removed, and this is followed by the addition of the 3A4 luciferin IPA substrate that Tracy described previously. And again, as mentioned, this is a cell-permeable substrate, so it can get into the cells, be acted upon by the 3A4 enzyme, and then the modified substrate can move back out of the cells back into the media. This facilitates the ability to remove a portion of that media after the incubation time to a separate plate, followed by the addition of an equal volume of the detection reagent, and then the luminescent signal is quantified, which is proportional to the activity of the CYP3A4 enzyme in that well. Back in the cell plate, the media is then removed, and you have the addition of the 3 millimolar salicylamide. This is done to ensure that any cleavage of the CYP1A substrate is performed solely by that P450 enzyme. So then after a short incubation period, the media is removed. You then have the addition of the CYP1A substrate, which is, in this case, we used resirufin ethyl ether with the addition of the salicylamide that's incubated. And then you have the fluorescent quantification of the resirufin that's created by the activity of CYP1A in the well. Following that read step, you have the addition of an equal volume of cell titer glow, incubation, and then the luminescence signal again is quantified, which, as previously described, that signal is proportional to the number of viable cells within the well. So again, to confirm that the multiplexed assay would generate the correct pharmacology results, we tested a number of known inducers of the two CYP enzymes in dose-response format. Fold induction over basal activity was used to determine the effect of the test compounds on enzyme activity, and we repeated the assay 3 times using the same hepatocyte donor to show the repeatability of the assay, and then the assay was also run using 3 separate hepatocyte donors to show the equivalent data could be generated with different hepatocyte donors. But before that, as with the previous case studies, we performed Z-prime experiments to validate the automated hepatocyte-based assays. And once again, you can see that the Z-prime values were in the 0.7 range, confirming that the assays were robust and delivered quality data. Okay, so here we show the induction curves and EC50 values that were generated by the two known inducers used previously in the Z' experiments, omeprazole for CYP1A and rifampicin for CYP3A, across multiple runs, again using the same donor and also generated using the 3 individual donors. And as you can see, the similarity in the induction curves and low variation in the EC50 values demonstrate the repeatability of the triplex assay. Furthermore, the close agreements between the EC50 values generated using the automated multiplexed assay to those generated previously in the literature demonstrate the validity of the process. Finally, the table at the bottom of the slide shows the average EC50 values and Emax values that were generated for the 8 compounds tested. And then the figure above shows, again, how all 3 pieces of information are put together to get a complete picture of the effects of each lead compound. The 4 compounds on the top row of the figure show induction by CYP1A and no induction by CYP3A. And the situation is basically reversed for the 4 compounds on the bottom of the graph. This is as it should be, since the top 4 compounds are known inducers of CYP1A, and the bottom 4 compounds are known to induce CYP3A. And they also basically don't show any induction of the other enzyme in the triplex assay format, except at very high compound concentration levels. In addition, some compounds, again, as was previously explained by Tracy, show induction of the enzyme, and then the induction decreases at high compound concentration levels. So again, by including that third piece of information, the cell viability data, you can see whether that decrease in induction is due to true inhibition of the enzyme at high concentrations of compounds, such as seen in omeprazole, or whether it's due to the compounds at high concentrations just killing off the cells, such as is seen in lansoprazole. So in conclusion, as I mentioned initially, higher throughput ADME-Tox chemistries continue to see increased emphasis in the drug discovery field. Small modular instrumentation allows for automation of the pain points of each assay, and microplate readers also provide a flexible solution for the detection modes used by many of these chemistries. And then finally, by combining the appropriate assay chemistry, model, and instrumentation, you can increase productivity and generate high-quality in vivo-like results in an in vitro setting. Thank you.
Okay, folks, we're coming up against our time limit here. So what I'm going to do is I'm going to try to get your questions answered via email. What will happen is the presenters will take the questions that you've submitted and attempt to answer them. You'll get those in your inbox. So I wanted to thank you very much for joining us today. Thanks to Tim, Tracy, and Brad for their presentation. And we will attempt to answer those questions. You'll get those probably within an hour or two. Be sure to check out our websites and blogs, like us on Facebook, follow us on Twitter to hear more about biotech webinars or learn about our products and how they can help you with your research. Have a great day.